For those of you who are genetics folks, you can take a nap for about 3 minutes. I do like to quickly go through some definitions for those of you who don't think about these things. And I get confused about, I used to get confused about several definitions. Oncogenes and tumor suppressor genes, we're talking about mutations in the genome. Oncogenes are genes that promote cell growth, and when they're mutated, you only need to mutate one of them, and they activate cell growth. And tumor suppressors, P53, P16, P10, et cetera, their normal function is to inhibit cell growth, and you have to mutate both of those, right? So everyone knows those. And then in the world, in the era of NGS and high-throughput sequencing, we now have layered 2 other types of mutations onto those, drivers and passengers. So it's important not to get confused that oncogenes doesn't mean driver. Actually, a driver can be an oncogene or a tumor suppressor. It just means it's an important mutation that drives the growth of that tumor. So it can be in either one of those. And with more and more sequencing, we think there are probably about 5 to 10 critical drivers per tumor in a full-blown malignancy. And then the opposite of that would be passenger. And so the more sequencing we do, the more mutations we find. Every tumor has DNA repair defects, so accumulates more and more mutations. That's generally true across the board. And so mutations that we find that don't change cell growth when we model them in vitro are passenger mutations. So the goal for myself, I'm a pathologist, I run molecular diagnostics, is to differentiate drivers from passengers, and that's the name of the game. What's the scope, if you stand back, and what's the scope of genetics? Let's say some people may say, I don't know, why don't we just do whole genome sequencing on everyone? Well, the problem is very sizable in the clinic. And so the human genome, the haploid genome has 3 billion base pairs, 20,000 genes. If you look outside of cancer, there are about 5,000 genes linked to any particular Mendelian disorder. About 1,000 or so, that's the number we've landed at. Everyone has their own different list of genes. Somewhere between 500 and 1,000 genes have been associated with cancer as somatically acquired. So obviously there are other types of variants, but this gives you the scope. It's a big problem that we'd like to reduce to practice, as to put it in Keith's terms. So before I go into some more of the science, let's talk about the regulatory challenges. There's also more definitions on this slide, which I think it's important for everyone to think about. Unfortunately, this is something we do in clinical pathology now have to think about more and more. So there's the FDA, there's CMS, there's the AMA, and then there's CLIA. And so all these acronyms. FDA asks one thing about devices, right? The FDA regulates drugs, but it also regulates devices. Does the device work, right? So you're selling a device or using a device, does it work? And they want clinical and technical validity. CMS is different, right? Medicare is different. And the payers are different. They want to know, should we pay you for doing that test? Just because the FDA said that, the test works doesn't mean we should pay you. And that brings up the issue of clinical utility, which is the main problem that we're facing right now, is no one has proved that broad-based genotyping, NGS, is clinically useful. I could basically stop my talk now. But that's just the truth. There's no argument to be made, and this is coming from someone who does this clinically. There's no proven clinical utility at this point. The AMA provides coding. You may hear of CPT codes that we use. The CPT codes are done using a molecular committee within the AMA. And then CLIA, of course, scares a lot of people. CLIA is a little bit different than the FDA, right? CLIA is more looking at the lab level and wants to know, does your lab know how to use that device, right? So as the FDA gets more and more involved in regulatory oversight of molecular labs, it's different than what CLIA is overseeing. And so CLIA asks us to do quality assurance and quality control within the lab. And so there's been a lot of press lately about lab-developed tests. Most of the tests we do, in fact, all of the NGS tests in cancer right now are, in fact, LDTs or lab-developed tests. There are no FDA-approved tests right now. Hopefully there will be in a year or so. And so the FDA is now looking at regulating LDTs much more closely. And this is a big deal, is a very, very big deal, because many labs probably could not withstand the costs associated with increased regulation. So happy to talk more about that if anyone wants to. And then the last thing is a little definition on the bottom. People talk about a CLIA-approved test. There's no CLIA-approved test. The lab is CLIA licensed, so that's just a little definition. The test is not CLIA-approved. So your lab, if you do it right, you can run the tests as long as you've done the right validations. Okay, so let's talk. I was talking with David Solid before, and he's mentioned that he's frustrated that a lot of people think that harm can be done to patients with kind of broad genotyping and genetics, and it can, it can be a danger to a patient if you put them on the wrong drug. But let's talk about the positive, right? I feel very strongly, and I think everyone in this, Keith and David, of course, would feel very strongly that this has been, if not the major, one of the two major breakthroughs along with immunotherapy in the last 5-10 years. And so crizotinib is something that our group knows very well. We set up the first FISH test for screening of non-small cell lung cancer for ALK rearrangements in 2007, right after Dr. Mano's paper was published. And so we began to do this before there was no FDA-approved test, right? We set up what's called an LDT in the lab, and we started screening every patient. We considered this clinical testing because, in fact, if you look at this patient here, everyone knows the responses in these patients are quite impressive. Our first patient, first 2 patients, first 3, first 4 patients, so really nice responses. And so Dave Ryan can comment, but this became standard of care at MGH within like 1 week or 2 weeks. And so when we had an LDT for a relatively unknown genetic alteration, but we had a drug in phase 1 for it, all of a sudden our clinicians needed this test on every lung cancer patient, and that's something we set up within a few months. Okay, that's great, but what's the benefit? Well, the benefit is if you do it right, in fact, you can go from the discovery of the alteration in 2007 to FDA approval in 2011. There are other examples, obviously, the development of BRAF inhibitors. Gleevec, of course, was done in this targeted manner, where you actually started with the genetic subtype, and then you did drug development later on top of that. But if you bring an LDT into the lab early on, I feel very strongly that it was the early launch of the LDT actually accelerated this. And it was those early few hits that allowed us to get the momentum to accrue the most number of patients. So I've tried to make this point to the FDA that we have to be very careful not to stifle innovation by over-regulating lab-developed tests. And you want the academic centers to be able to do things like this, obviously with patient consent, knowing that the real risk here is not the test, the real risk is getting the drug, as long as the patient's consented right. So I think early development of tests, early implementation, allows us to do clinical innovation in a way that really help patients. And for ALK, I would argue, and I think we can do the economics, that there's probably a year acceleration for which a year ahead that crizotinib was approved that we couldn't have done without that. And you just think of the number of patients that helped. And so if you're a naysayer, I think there's also the flip side that we can do this. So what are the considerations in clinical panel testing? So in 2007, 2008, we started looking at larger panels of genes simultaneously. I'll go into that in a little bit of detail, but if you want to start doing panel testing and not just ALK alone or EGFR alone, there are a lot of issues that we have to deal with. We have formalin fixed paraffin embedded issues, analytical sensitivity, turnaround time. David will talk a little bit about what they're doing up at Memorial. There's a mix there of clinical and research. We're doing all of our testing as clinical testing. We bill insurance. Logistics is not a minor component to be able to turn around tests in a reasonable amount of time. Tracking requests and patients has not been straightforward. Actionable targets, we can have an entire symposium on people debating what's actionable and not actionable. Does it predict response or resistance, diagnostics, and prognosis? Are we just looking to stratify patients for trials, et cetera? So we spend a fair amount of time on that. And then for me, as I've been talking about so far, is sustainability. So to make this sustainable in the clinical environment, you have to show it's economical, and then you have to convince insurance and billing. So you have to show clinical utility. I would add new NGS CPT codes are being launched now, and the question remains, will anyone pay even if we have those? So let's move through those. I'm not going to talk about SNAPSHOT much, but this is the assay we launched in about 2008. At the time, it had 24 cancer genes, a hotspot mutation assay, and it served us very well. I'll come back and show you some other evidence now that we're using NGS that actually this panel is quite cost effective, and as a first-line screen is really not such a bad idea. We get a lot of the major mutations that we're interested in. What did we learn about it? Well, first, just to show you, we tested basically all tumor types. We performed these tests at MGH SNAPSHOT and now NextGen at the request of the clinician. We don't reflex every patient that comes through the door. And I think that's, for a lab that wants to do this in a clinical sense, that's absolutely necessary and protects us from a lot of patient consenting issues. So we do almost every tumor type. And then this is probably the most important slide. You don't need to look at any of the details other than to know that these are the anatomic locations of the tumors and these are the genes that are mutated. And every red bar is a percentage within each gene, what percentage of mutations are found within each tumor type. And almost every gene in every tumor type has some percentage positivity. So I like to show this slide. Again, you don't need to know or look at the details, but I strongly believe that we don't need disease-specific panels, that really one assay is going to provide useful information about drivers in almost every tumor type. So BRAF is a great example. David will talk about it. We find BRAF literally in every tumor type at a certain percentage, and so this is important. Obviously, NRAS, AKT goes down the line with those. What else did we learn? Well, this is data from our SNAPSHOT assay. SNAPSHOT, again, being a hotspot assay, had a large percentage of cases we found no mutation. This is the prime reason to move to next-gen sequencing. But, you know, we didn't help everyone that came through the door. 40% of patients didn't have KRAS or EGFR. The other thing we learned is some idea how to deal with these rarer mutations. And rare mutations are really, really interesting and really, really challenging. It brings up the question is how much time and resources do we want to spend to find any given particular individual? And to me, slides like this show absolutely that we have to do large-scale panel screening. There's no way that you could do an individual test for KRAS, then EGFR, then ALK, then ROS, then RAS, then RET. There's just no way. We run out of, I mean, for no other reason than we run out of tissue. And the cost to do this, by the time you add KRAS and EGFR and ALK together, actually an NGS panel can already be done for less. So this slide tells me one thing. Panel testing is the only way forward. It tells, well, it hints at something else that I think David will talk about extensively, and that is how do you do trials of things that are less than 1% of a population or less than 1% of a population? And so basket trials that I know David will spend some time talking about. Dr. Flaherty is the overall PI of the NCI match. This is an interesting type of basket trial, an umbrella protocol, so to say, multiple phase 2 trials. Keith is really leading the clinical implementation of this and the collaboration with pharmaceutical companies to find all the agents. We are using a next-gen sequencing test from a commercial company, from Thermo Life Technologies, Ion Torrent, that will be tested at 4 CLIA sites. And then patients recruited through the cooperative groups and academic centers. And the hope is that not only will we'll be able to move drugs forward in the proper genetic subtype, but we may actually be able to help move forward an assay for FDA submission. And then hopefully if we had FDA approval for a test, we might get paid for doing some of the work we're doing. So I love the idea. I applaud Keith for helping push this through, and I certainly applaud the NCI for doing something that I think is very innovative. Right? I just think this is really innovative, and it's really shows someone in the government trying to help, you know, shake the system up and do something to move forward. So we're thrilled to be able to at least participate and help along that. A lot of lung adenocarcinoma. This is a summary of some of the work from the Broad on genetic alterations in lung adenocarcinoma using exomic sequencing. Again, don't need to know the details. The usual suspects are up here. One thing I do like to point out is that, you know, there aren't thousands of mutations here, right? There's commonly recurrent mutations. There's probably only on the order of 30 to 40 genes that are commonly mutated. If you talk to Gadi Getz over at the Broad, he'll tell you that, okay, some of these are rare, but that's, you know, these aren't that rare if you sequence more. And if you continue to sequence another 1,000, 2,000, or 5,000 samples, this list will continue to grow and grow. And so the tail on the low prevalence mutations will continue to rise. And then copy number is shown down here, and I think we have a reasonably small number of copy number mutations. So the problem is sizable. If we want to comprehensively sequence, let's say, all lung cancers, we have to sequence probably most or all of those genes in their entirety. And how are we going to do that? So this session is on next-gen sequencing, and so I should at least have one slide talking about next-gen sequencing. For those of you who know all about it, just ignore the slide, but the amazing technological advances over the past 5-10 years, led largely by Illumina and Life Technologies, have the ability to generate huge amounts of data. Now within just a few days, the 8-day runtime is the HiSeq 2000. You can do it within about 2 days. We have several challenges. There's huge amounts of data. And so for a clinical operation to deal with huge amount of data is not that easy. Several terabytes of raw image data. We don't keep that, obviously. But quickly we fill up terabytes of data within our storage servers. And this is just an area of the hospital that no one's really been prepared for on the clinical side. Even the imaging folks, the radiology folks, use so much less data than what we're talking about in the genomics era. So the exciting news is we have the ability to do huge amounts of high-quality sequencing very fast. The other side is the challenge on the bioinformatics and the data storage. So I feel pretty strongly about clinical genotyping and the type of platform that we should be using, at least for the foreseeable future. You can argue, some people again may say, let's do whole genome sequencing. Like, why are you bothering with panels or anything else? I have, my family member has cancer. I want to know everything. Let's sequence the whole thing. Well, it's still costing $5,000 to $10,000. Many of the first steps in analyzing whole genome sequencing is to eliminate the non-coding region. There's a few left. Otherwise, there's just too much data to analyze. So if the first step is to just drop out all the non-coding regions, then exome sequencing is probably the one thing that would be somewhat reasonable. It's not that expensive if you think about the big picture where we are in terms of pricing of tests, and commercial next-gen sequencing tests being on the order of $5,000 to $6,000. $1,000 to $2,000, kind of reagent cost for an exome is really not too bad. I would argue the analysis is still complex and the turnaround time is really too long. And that's true of whole transcriptomic sequencing if you're looking for fusions or RNA alterations. So you can argue about it, but I would say right now we're still not in an era where these are really practical, and certainly not from standard FFPE material where you may be only getting very little. And so you've probably seen this slide a number of times, which is from Gaddy's group at the Broad, which shows just the mutation burden for a variety of tumor types, melanoma and lung cancer, those diseases associated with high mutation burden being up here, and pediatric tumors with little DNA damage, chronic DNA damage shown over here with having few mutations. And why am I showing you this? Well, the issue is, from a clinical interpretation standpoint, is how do you interpret one of these lung cancer exomes? And so we did a study of 4 whole exomes for lung cancer. This slide is showing you smoking pack years here, sorted by the number of alterations, SNVs and indels. And you can see that most of the patients, at least the smokers, have somewhere between 200 and 1,000 variants. This is after filtering out SNPs and whatnot. So as a clinician who has to interpret this data, it's nearly impossible to interpret everything. You have to make some assumptions to whittle this number down to a reasonable number to interpret. But it's really a huge challenge, and I think everyone thinks they have an approach to it. There are some commercial vendors that have approached only interpreting kind of standard of care variants, for which there are probably 10 genes that have kind of standard of care genotypic interpretations. But if you're looking at 500 mutations, it shuts the lab down, basically. The other interesting thing, just a little sidelight here, is I highlighted in red the non-smokers, just those guys that are down here. You wouldn't be surprised that the non-smokers tend to have a lower mutation burden, right? So the ALK patients are here, the ROS patients are here, some of the EGFR patients are down here. And this study was really interesting for another reason. There was one non-smoker up here, right? And so one interesting thing, especially from a pathology standpoint, is NGS has the ability to correct some diagnostic errors. And so this was really weird. The patient with the most mutations was a non-smoker, and this was a lung squamous cell carcinoma. After we did this research project, I contacted our clinician. I said, do you know anything weird about that patient? Is there anything else in the history, any other tumors? And in fact, the patient had a smooth and mutant basal cell carcinoma of the skin that was locally highly aggressive, had been radiated multiple times. It was called by the pathologist a basaloid squamous cell carcinoma of the lung, basaloid-looking squamous cell. But in fact, this was a metastatic, and we confirmed that by sequencing both of it. So NGS, in fact, this is 1 out of 40. That's, for pathologists, that's a bad error rate. And so I think we're going to have the ability to do some interesting changes to diagnosis. So let's compare the different assays. I hope I've convinced you. Exome and whole genome are probably unwieldy in the clinical lab, but they offer something that panels don't offer, and that is a huge amount of discovery potential. So in an academic institution, we have probably equal balance of clinicians here. Dave can confirm this. Dave Ryan can confirm this. Half of them don't want any more information. They want the minimal amount of information because they don't have the time, perhaps the understanding to deal with it, and the other half want the entire genome, and why can't I have it tomorrow? I've tried to convince everyone that hotspot assays make a lot of sense in the clinic right now, since we're not getting paid for nearly anything that we're doing. Being able to do things with low sample input at a very low cost with a reasonable turnaround time, we would like to have it in 1 to 2 weeks, with easy informatics and interpretation is probably the way to go. Specimen size, if we talk about solid tumors, specimen size is a real big issue. 40% of our samples are biopsies or cell blocks, and that's our samples. Since we don't test everyone, we get very few early stage tumors. We're getting largely metastatic disease. 25% of these have less than 150 nanograms total of DNA in 10 5-micron sections. So this is a real challenge. We have to use FFPE. I don't think using fresh material makes a lot of sense. So this is the type of sample here, a reasonable size resection. These are easy to deal with, and this is a core biopsy that we get very little material on. And so we were finding that about 20%, as we were developing our larger next-gen panels, we found about 20% of the patients we cared most about, those with metastatic disease, didn't have sufficient material to be tested. So we developed a smaller focused panel that we call SNAPSHOT NGS, or a snapshot replacement assay. For those of you who are aficionados of NGS, I'd be happy to talk about it. This is a home brew assay, an LDT, so to speak, using a technique Zhengli Zheng, my postdoc, developed, anchored multiplex PCR, which is similar to RACE. It allows us to look at about 40 genes, which may not seem a lot, but it really gets you the majority of information that you may need, with some bonuses. The bonuses are that we now have very high quality sequence based on this technology. It can be done quickly, cost effective, less than $200, with 5 to 10 nanograms of input material. Right, so do you want a big gun $5,000 assay, or would you like a well, not that rapid for us, but potentially rapid assay? And the good news, I would say, is that we're getting more comprehensive. Instead of 60% of samples having a detectable driver, we're now over 85% of lung adenocarcinomas. And I do like to make one point. I've heard Matt Meyerson make this point before, that KRAS is really common. Right? So the reason to point that out is, one, everyone should really be focused on KRAS. I mean, I'm going to talk about ALK and ROS. I really shouldn't talk about that. Right? Everyone should just be working on KRAS, and I think the NCI knows that. I think everyone knows that. But this is a run from yesterday. Actually, I signed out 18 cases yesterday. These are all different tumor types. This is not just lung or colon cancer. There was an ovarian case in here. There was an AML in here. And the first 5 cases, I was putting this talk together and signing out, I was like, KRAS, 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 KRAS. So do you really need to spend $5,000 on all of those to do deep sequencing to find KRAS? No. And so when over 30% of the cases in one run, I really think there's a cost-effective, rapid approach to doing this. Without belaboring the point, I think there are other technologies than NGS that are good for rapid turnaround time. For BRAFID melanoma, IDH in brain, we actually have mutation-specific antibodies, which pathologists really love. I really like those if we can improve the sensitivity and specificity of those. So I think there's a way to improve the speed, at least for the most common things, and then you would go to a big, expensive gun if those are negative. So we also have a discovery panel, which is still in the research development mode, which is our 1,000-gene panel. This is the assay that we slowed down development as the situation with payers degenerated over the past year. So the cost is just much higher. There's a linear response to the number of genes you look at and the cost of sequencing. And so if you're looking at 40 genes, it's just much, much less expensive for sequencing. But this has the ability to do some discovery from FFPE material, and we can get down to about 200 nanograms of input material with that. Okay. I'm going to transition to fusions and fusion detection. That's really where we spend in our group most of our efforts on R&D, and certainly is trying to build on the work that we've done clinically with Eunice Kwok and Alice Shaw on developing crizotinib in fusion-positive cancers. So detecting rearrangements, there are a number of ways to detect rearrangements. IHC, I mentioned, is really good for mutations. It can be really good for fusions, and ALK is a great example. ALK is not normally expressed in lung, so it's a beautiful assay. If you have ALK expression, the ALK kinase domain is expressed, you basically have the fusion because there's no normal ALK in lung, and so non-fusion negative lung cancers don't express ALK. That's not true with ROS and some of the other fusions. So about 30%, for instance, about 30% of lung adenocarcinomas express relatively high levels of ROS. So we're not going to be able to use a ROS antibody to be specific for the fusion-positive cases. So IHC has a place sometimes. FISH is something I've done a ton of, built kind of our lab and a lot of the clinical stories around FISH and the development of FISH. It's a good assay. It allows you to see single tumor cells and make calls on single tumor cells, which is really powerful. But it's technically challenging, and it's not scalable. You can't do 10 different fusion genes at the same time, and you run out of tissue. Again, we have to balance not running out of tissue. RT-PCR obviously is a great technique. RNA from FFP can be poorly preserved, and also you need to know both partners. So those two major limitations pushed us down the NGS route. And so why would you need NGS for fusions? Well, I can just show you a few examples. ALK fusions. This is pulled out of Cosmic. These are just the fusion partners for ALK that have been previously described. Obviously, EML4, wherever EML4 is up there. NPM1 ALK in anaplastic large cell lymphoma. But there are a lot of partners, and each of these partners might have multiple breakpoints. So if you're going to do RT-PCR, you need to know both primers. You already have probably 30, 40, 50 primer sets, maybe 60 primer sets. ALK-FISH, as I mentioned, is a little bit difficult. The two genes are very close together, and so when you're looking for a split signal, those are actually really challenging. And a lot of commercial labs I know have had trouble with this and called a lot of false positives. So I hear that from Alice quite often. One interesting thing from a lot of the TCGA and NGS data being published is a lot of the new fusions being discovered. Actually, the genes are very close to each other, and that's because they've never been visible in prior cancer research in karyotyping or other types of analyses. And so it's only now that you're sequencing that you begin to see things that were formerly invisible to kind of cancer cytogeneticists. And what that means is a lot of the new fusions actually aren't amenable to FISH analysis, because when the genes are too close, you actually can't do kind of standard split apart analysis. So FISH is becoming less, in my opinion, less important. So we have all the different ALK fusions. ROS obviously was discovered actually about the same time as ALK fusions, surprisingly, but it took us a little bit longer to catch up in terms of clinical development. We've used FISH for the first 4 years or so, but have now transitioned to NGS. And why do you care? Well, if you find the right ROS patient, as you've seen with ALK patients, you can really show dramatic responses. For those of you who don't know much about ROS, it's really more like 1% of lung cancers. And so when we use FISH, you have to look at 100 patients to find one, right? And just, it's a lot of money, right? FISH, although not an expensive test, if you're doing 100 FISH tests to find that one patient, you're going to break the bank if you have to do RET now on every case and you have to do ALK now on it. So you're doing for 100 patients, you're doing 300 FISH tests, maybe to find 4 patients. And if you're this patient, did it make a difference? It's crazy. Right? It absolutely made a huge difference. And so we've always felt it's worth putting the resources on these type of problems, but it's not really scalable. And as the number of fusions goes up, our oncologists get angrier and angrier that why aren't we keeping up? Well, there's only so many hours in the day that you can do it. So that's ALK and ROS. Just in the past year or so, NTRK1 rearrangements, a huge number of FGFR 1, 2, and 3 fusions described. And the list goes on. I'll come back to those. So we had an unmet need, we thought, for an assay that didn't need to know the 5' partner. So we developed this assay. Again, Zhong Li and my group developed this technology, which is similar to RACE, where you put on adapters to your library. You have a double-strand cDNA library here. and you use 2 gene-specific primers in a row, 2 nested PCR reactions. And your adapter PCR primer is non-specific. It doesn't bind to any specific gene, so you actually only need, you do this PCR reaction, and you only need to know one of the partners. So you'd only need to know, for instance, the ROS kinase domain, and you lay down your primers in the ROS kinase domain, and then you face those primers pointing 5'. So any possible fusion primer you would be able to detect using bioinformatics. We read out of each of these exons, and then we read towards the 5', and if we find something novel here, our bioinformatic pipeline will be able to say, okay, you didn't see the next ROS exon, you saw a gene you didn't expect, and then it tells you what is that gene, and it tells you is it in frame or not. And so it's just, you know, it's just a very simple and it's been effective assay. We've completed assay validation over a year ago. This has been our standard fusion assay for over a year now in lung cancer. I won't go through all the validation studies other than to say it performed really well against ALK FISH. So we did really well for the positives and negatives, and actually managed to classify some ALK FISH questionable cases. I won't go into details about that, but FISH can result in some indeterminate cases which are put into a positive or negative bucket by that. So some interesting fusions. There's been a lot of interest in the past few months in a subtype of lung cancer called invasive mucinous adenocarcinoma. Some novel fusions. We actually started working on this with fellow Karin Finberg and Licia Sequist back in 2007. This was my first publication as a staff member looking at mucinous adenocarcinoma. We found these mucinous subtypes that are very specific appearing tumors. They're interesting. They're filled with intercellular mucin. They look almost like goblet cells of the colon, and they have basally located nuclei. Their pathologists picked this up right away. And so one thing we noticed is when we were testing for EGFR in this era that these were all EGFR negative. And that was the only test we did. We started doing KRAS and found the majority of these were KRAS positive. In our cohort, almost all of them were smokers. Roman Thomas's group from Germany just published this past year, however, that there's a really interesting novel fusion that's detected in a large number of these KRAS negative mucinous adenocarcinomas. These were all female never smokers, so another fusion that's arising in a non-smoker. Neuregulin is a very interesting gene that binds to ERB3 and activates HER2 signaling. And when it's fused to CD74, it becomes kind of constitutively membrane bound, and it can activate signaling. So it's a really neat signaling molecule fusion that's novel. It's about 20% of mucinous adenocarcinomas. And then the Japanese group followed soon after with another paper showing RET fusions, BRAF fusions, ERB4 fusions on top of neuregulin fusions within this mucinous subtype. We were scooped by both of those papers, but also have, out of 16 KRAS negative cases, had 6 novel fusions, including 2 different neuregulin fusions, BRAF, NTRK1, and ALK. And so why do we care? Okay, so you're finding more fusions. Well, these are all targetable, right? Every one of these has an agent that Keith can come up with a rational approach today or tomorrow, something that's already in trial. So it's a very interesting subgroup. Fusions, I don't think we've reached the end of the fusion discovery. The Blueprint Medicine group from Cambridge actually mined the TCGA data. The TCGA didn't publish these fusions. This was the public TCGA data. Blueprint went in and just mined the data looking for novel fusions from the RNA-seq data, and they found a huge number of interesting and novel fusions listed here, and this was just published a few weeks ago. So two lessons. If you're interested in discovery, bioinformatics of the TCGA is probably a good place to get some Nature communication papers. And two, the growing list of fusions is really quite remarkable, and I think these are incredibly targetable. I mean, they're clearly important drivers, and many of them are the principal driver in these tumors. So actionable rearrangements in lung tumors, you know, if you talked to me a year ago, we'd have ALK, ROS, and RET maybe as the new one on the block, but just within the past year, we've added all of these other fusions. And so the percentage of them, none of them are really, really common. ALK would be the most common. They're all in the 1% or less range. You add them all up together, it's probably no more than 10% at the absolute highest. We don't yet know about the non-smoking status of these other fusions, but it certainly is interesting that the majority of fusions are arising in non-smokers. So there's something that we don't understand about the epidemiology of these non-smoking tumors that I think is a fascinating area. The last thing I'll point out is there's a number of protein kinase A subunits, cyclic AMP signaling pathway subunits here. Also in some liver tumors, these have been published. So There's a lot of interest I see percolating around that pathway, which is interesting. CNS tumors are another area we look in. There's a growing list of similar fusions, actually, in brain tumors, and I think this is going to be true in all kinds of different tumors. So we're finishing final validation. I've been saying that for about 6 months to a year. Of a fusion discovery assay, which is on the order of 50 genes now. So we'll be able to look from FFP materials for all of these gene fusions. In the last few minutes, I'm actually going to transition back to kind of the more mundane day-to-day challenges of doing NGS in the clinic. So customer support. This has been a challenge, in my opinion. I think sometimes we've done a great job helping our clinicians, and sometimes we've under-delivered. There are some major challenges. Some oncologists will not want the data for larger panels. And I would say the good thing about MGH, they don't have to get it, right? People who don't want to order the test don't need to order the test. Often we will not know what to do with the data. One of our biggest challenges in interpretations are variants of unknown significance. So the more sequencing you do, the more variants of unknown significance you do. The challenge with variants of unknown significance is sometimes we don't know if they're germline. And how do we, I always get the question, how do you deal with germline variants? I don't think anyone has the right question for this. We do refer some of these patients to, if the alteration is clearly deleterious, we do refer some of these to genetic counseling. But we currently have not solved, in my opinion, we have not solved this problem, and we're just at the very tip of the iceberg in dealing with this. The other thing is, I think we have to be very careful about what we're doing. I think certainly some people think that this type of analysis is putting patients at risk from a drug risk standpoint. But I think we also have people who think that we're just out trying to make money, right? And this is a money-making venture rather than doing something that's trying to help people. So we talked about variants of unknown significance. I just pulled this slide together. We've done a lot of NGS of pancreatic cyst fluids. This is different than predictive genotyping. This is diagnostic testing. We're looking for KRAS mutations in neoplastic cysts and GNAS mutations in neoplastic cysts. These were 4 cysts that we got definitive diagnosis of, but at the time we did SNAPSHOT. We were looking for KRAS and GNAS. We found mutations at around 50% allele frequency in these interesting cancer genes. So every variant we find is actually in an interesting cancer gene, because we only look at interesting cancer genes. So these cases cause great consternation amongst our gastroenterologists and our surgeons and our pathologists, because these were not previously described as SNPs. There was nothing in the literature, nothing in DB-SNP or any of the databases that said these were known SNPs. Their allele frequency suggests 50%. Really smells to me like a heterozygous SNP, but we felt like we needed to report them. And so you're stuck with a patient. Do they have an APC germline variant, for instance? And could they have, could they have some germline alterations in P16 or NRAS? So when you're doing diagnostic testing, variants of unknown significance actually get much more stressful, because if you were to say these are deleterious somatically acquired alterations, that would make the diagnosis of neoplasm, and that patient may go to surgery. So in the diagnostic realm, these are very, very scary. In the predictive realm, where you're trying to decide should the patient get some MAP kinase pathway inhibition, I would say it's less stressful, but equally dangerous. The other challenge we have long-term is displaying the data. So with the small panel, it's relatively easy to display data for small panels, but as we move to larger 300 or 1,000 gene panels, we've actually spent a lot of time behind the scenes working on a genomic data workflow. And to be honest, a lot of the tools necessary to manage this kind of genomic data long-term in the clinic is absent. It just doesn't exist. There's neither commercial or in-house tools to do that, and that includes tools that manage the LIMS, that is the lab information management system, just the specimens coming in and onto instruments. Obviously, we have a huge amount of time and money, and David, I assume, would say the same thing at Memorial on computational biology and bioinformatics. Gary Getz and Long Lei in our group lead this effort, and this is a huge effort with a lot of really, really smart people. But really, really smart people don't necessarily make great great clinicians. So I don't know if that goes both ways, though. Most clinicians aren't really, really smart. We need to store the data. And again, storing data has actually become a huge expense. And then the biggest challenge, and I think David Solid will agree with this, is the genetic knowledge base. Building a set of interpretations that allows us to analyze data quickly rather than to start over on each particular case. And so what we'd like to build, and are currently in the middle of some pilot projects, are building a kind of dashboard. We soon will be an Epic shop, so this gets to be a little bit of a challenge doing anything innovative on top of the Epic system. But to build dynamic reporting and intelligent analytics dashboard that can respond to new clinical trials that Keith publishes, for instance, in real time, I think is the way of the future, as opposed to a static report where on that day you've got these variants with that interpretation done. I think eventually clinicians will be logging into a dynamic reporting database where their patient's genotype will be updated in real time. How to interpret is a challenge. Rodrigo Dienstmann was a fellow in the lab, put together this for us, which to me was really, really helpful in how we think about in the clinical lab, how do we report and how do we organize our thoughts for clinicians. And so overriding this is what is reportable. Underneath that are actionable targets, other relevant variants, and then unknown. Unknown is really easy, right? That's easy for us. We can just dump unknown into there. And consensus is really easy. Everything else in the middle is really hard, and emerging is really where we're struggling. So all the commercial solutions that exist for this type of analysis are really using consensus guideline documents. None of that's a big challenge. We can handle that internally. NCCN or FDA approval documents. Emerging is tough. What kind of data do you give your clinicians? Do you give them preclinical data that one of your researchers has really exciting animal model data suggesting that drug might work in that genotype? Or do you wait for late clinical trials? Prognostic and diagnostic information, there's a huge amount of data that exists out there around just pure prognosing and diagnosing tumors. In fact, most papers published in cancer, like clinical cancer research type papers, are these type. They're not necessarily predictive. And so if you just look at P53, for instance, and you pick your favorite tumor, there's probably 1,000 papers looking at the prognostic significance of P53, and I would bet half of them say it's a positive and half say it's a negative prognostic indicator. So these are very challenging. You need very strong evidence to report these out. And other relevant variants we won't go into, but things that are clearly of biological importance. Can we leverage genetic and clinical data? I think it's really important that we try and share data across institutions. I know there are multiple efforts at the NCI level and other levels to be able to do this for no other reason than to find sufficient number of patients for trials. I think NCI Match will have this kind of infrastructure. I've heard some really interesting things coming out of Medicare to build this type of data sharing infrastructure. The other question I always get is about tumor heterogeneity. Naysayers will say, well, tumor heterogeneity is so complicated that targeted therapies are really never going to be that successful, right? Because the tumor can always avoid. So I do have one just informational slide, and these are my opinions only. These are not official opinions. These are mine. Because it comes up so often, point mutations can be heterogeneous within a tumor. And so initiating driver mutations, things like KRAS and ALK and ROS and EGFR, in my opinion, in my experience, are not heterogeneous. And Leisha Sequist in our institution has biopsied, I would say, 150 or more EGFR positive lung cancer patients following relapse on an EGFR inhibitor. Every single one of them at relapse has the same original mutation. I think David Solid, he published some interesting paper a few years ago about KRAS in colon cancer, metastatic colon cancer. They all, if I remember, you did a beautiful job showing they all had KRAS mutation when they metastasized. So in general, not heterogeneous. What I would call progression driver mutations are often heterogeneous. So you get a second PIK3CA mutation or a beta-catenin mutation. So if you're looking for the initiating driver mutation, you think that's your main target, you don't have to be stressed out that this is the wrong drug and you're never going to do anything meaningful. Gene amplification is a completely different story. And I would say there's two types of gene amplification events. The homogeneous staining regions, those are stably integrated. HER2 is most often of these type. Every daughter cell acquires that amplification. So these are fairly stable within a tumor. And then double minutes, EGFR and GBM, and make a neuroblastoma, these are present on extra chromosomal plasmids. These are always heterogeneous. So every single tumor cell has a different number of these. So anyway, that's my information slide to end up. So to summarize, we did a lot of work on fusions and a lot of NGS work on fusions. Preserving clinical tissue, I think that would be my main pathology point. NGS panel testing, in my opinion, has replaced other molecular assays as standard of care. We just need to convince the payers and the FDA that that's the case or should be the case. And just to thank our group, especially to thank Dora, Long, Darrell, and Leaf, who've been leading the charge for a number of years, Gaddy on bioinformatics, and Zhong Li, who led the development of the NGS technology. Thank you.
The first is a love letter, John, wanting to thank you for your always more than accommodating receipt of challenging cases from those of us who are out in the colonies wishing to get your opinion and your help, and I want to thank you for always for that. And the second is a question, and it's also sort of a question dealing with informed consent. The CEO Roundtable, and Bruce can speak to this because he was at the recent meeting on Friday, has, as you may know, John, the last several years been working on data transparency and data sharing of clinical trial data. Yeah. And this data has got to be de-identified before it goes into the sphere, and when it is there, then it's freely available to everybody, including SAS Analytics. And Bruce raised his hand, and what did you say, Bruce? What about genomics? We need to have these profiles. And the problem, John, in this, I'm looking to you for a solution, is that the more genetic information we have, the more genetic information we have, the greater challenge it is to de-identify to that point. So are you getting, when your MGH patients are coming in and they're getting now, whether it's SNAPSHOT or whatever, are they giving you informed consent to use and that data in a transparent fashion? That's my question.
I think it's a great idea. It's not something that we've done yet. We've talked for many years about getting a blanket IRB consent form. Our consent form is purely a clinical consent form for this type of testing, which is useful. It doesn't help us share data for research, so we need a separate, completely separate IRB. One nice thing about going the clinical route is that it's in the clinical sphere, it's under regulations of HIPAA and everything else, so we're really protected to put it into the medical record, et cetera. But for research, we need a specific IRB to share and look at that data. The question, which is the million-dollar question, it's probably a billion-dollar question that you brought up, is genomics is identifiable. And so you may say in isolation a person's genome is not identifiable. Well, someone may have the key to that genome somewhere, right? Someone may have a blood specimen somewhere. And there's never been an example of something bad happening in this space. But I think it's a matter of time before we read about something in the New York Times or the Boston Globe about something going wrong with genomic identity being revealed in a bed. I mean, there's laws, there's GINA, there's laws to protect individuals. But I have no answers. And the reason, I don't think anyone does right now. Everyone is so stressed. You know, I think standard clinical information and HIPAA stresses people. But people know what that information is. They kind of know what would happen if all of your record got out. You can appreciate that. Nobody understands, like deep down, what happens if, you know, someone in Russia gets your genome, right? It's kind of an unknown about the risk level. And for me, that's what makes it so much more stressful. Not everyone agrees with me, but I think that's strong. I would just add the last thing that we're trying to do right now, which we've reached this roadblock, is using the cloud for some of our bioinformatics. Like some of the tools we have, we really need to get stuff into the cloud for all these cool tools and the ability to do cloud-based analysis. And we're kind of at a standstill, because how can we be sure we can protect this kind of information in a cloud environment? So we've reached this point from many directions, and I'm not sure who's the right person who can say it's okay to have that be at risk. So great question.
Let me just add, though, to underscore the point about the benefit of hospital-based clinical testing. which is that the IRB consent needed to a protocol/consent to be able to then enable research, I mean, population-wide, at least, you know, at an institution, is a chart review because it's in the record, right? So it's really not, I don't mean chart review that you get the medical student to go through chart by chart, but I mean, it's really, you know, database exploration becomes a matter of chart review. And then it just allows you to be able to leverage all of the benefit of the kind of protected environment that is the hospital with its overseeing IRB. I mean, just it avoids some of the sort of fright of, well, this is, you know, NIH-funded research, has to go into a public repository, then what happens? I mean, when it's in the clinical record, it's protected along with the rest of the patient's health information. So it does kind of wall off some of that concern. Oh, sorry.
So this, you mentioned the NCI trials that are going on now, which are genotyping huge numbers of tumors, potentially a really valuable database for others, both the control arms of some of the studies and the treatment arms, because it'll tell you, you know, very large population of patients, what's the frequency, what kind of translocations you'll find, and mutations. Is there a plan to make that data available?
Keith would know more than I.
Yeah, well, John and the 3 other sequencing centers will be producing the evidence, but it's, yeah, no, that's absolutely, it's built into the plan. I mean, this is an NIH-funded trial from the beginning, so you can imagine that we're living by the rule that this all become publicly available, you know, de-identified from, you know, an individual patient perspective, but at a population level, definitely pooled data will be released. One of the points that John raised across a few slides is just this issue of finding somatic alterations and the co-occurring alterations with them. That alone, I think we can help with the MATCH trial as we, you know, characterize all variety of common and rare cancers. Just finding a fusion in FGFR doesn't necessarily tell you that it's important in that, you know, rare subtype of sarcoma in which you found it. But it helps if you then look at the rest of the actionable genome and say, well, it's clean otherwise, it just has that. You know, that type of information would give you, you know, some useful enthusiasm for thinking forward, not just for that one patient in the MATCH trial, but then following it up with further investigation. So no, all of that, you know, what I refer to as clinical TCGA. It's not as deep as the TCGA, but still, it's, you know, clinical patient population looking for therapy in clinical trials, which is not what the TCGA is. Will give us a better representation, I hope, of oncogenes in a real patient, in a real clinical practice population. Matt?
So I was hoping to get your thoughts on sort of what I have always, as an outsider, always sort of viewed as the elephant in the room around these things, which is the notion of how do you know what the driver mutations are? And I think this is implicit in sort of, you know, as you sequence lung cancer, you have this big 40% chunk that you're not sure what the driver is. And I guess I'd like your thoughts on sort of two things. One is I sort of see the biggest value of these efforts as gathering data, sort of the research question about can we figure that out, which has its own implicit challenges. Yet I often see the marketing is pushed as we'll find the rare variant that will then offer you therapy. And I think, you know, there's a disconnect there. And I guess I was wondering if that is that a product of the regulatory and sort of, you know, logistical environment, or do you think there's appetite for the discovery part? And then the second part is sort of speaking to that 40%, do you think it's going to be made up of 60 less than 1%ers of kinases and other things that we know how to call as oncogenes, or do you think we need to spend more and think about how do we find the variants and things that are not driver mutations in any way that we know how to think about today?
I think your first point is really important. I think we're doing both. We're both doing research and we're trying to find that important variant to treat that patient. And I think it's probably wise if people think that that's what we're doing. We're doing both at the same time. And for people to take either of those, I think harms the field, because the people say, you're doing research, forget it. Why are you billing insurance? That's research, stop doing that. And the other side are people saying, we're going to cure people, and actually you're not curing that many people. And so that makes people depressed and think that we're wasting time and energy. So we're actually doing both. I'm somewhat reassured. I had some interesting conversations with one of the medical directors from one of the Medicare carriers that there may be a way forward, something called in Medicare like payment with coverage with evidence development. and that Palmetto may be willing, Palmetto is one of the Medicare carriers, may be willing to move forward with exactly what you just described, right? Knowing that the government is paying for something from their clinical coffers that is partially to develop data and information. So as long as we have both of those in mind, I think we'll do great. I'm sorry, the second question?
If it was more your thoughts on whether or not the unknown drivers are the less than 1% stuff, or if it's...
I probably should let David get started. After he's done, you may not have that question anymore. But there is some dark matter left, right? 10% of cases in the TCGA where we don't know what the drivers are. I feel pretty strongly about the genetic basis of tumors. There might be a handful that are kind of epigenetically driven, purely, something like that, really interesting. But most of them, I think, would have an underlying genetic alteration. Some really interesting data about enhancer fusions, bringing enhancers closer to MYC or something like that. There could be this kind of dark, unknown genetic mechanisms, which are really based on a chromosomal rearrangement or a mutation, just a mechanism we've never picked up. So I'm really interested to see how that plays out. But I generally think almost all of them are going to have a genetic basis. Okay.
Was the CD74 neuregulin 1 fusion actionable?
Yeah, so you could come at that with HER2 inhibitors, for instance, or B2 inhibitors. Because if you presume you're inhibiting that pathway, you could come at that with a number of logical approaches. No one's proved that, right? No one's proved that yet in patients.
It wasn't done with that patient.
Not yet. Bruce would know some of the trials that are being developed in that area. So I would say we'd hope to know, and David may know. He's smiling.
Somebody let me know. I'm really interested in that.
Okay.
My name is Mike Shepard. Let's see, the other question I had for you, the ALK diagnostic was developed super fast.
Yeah.
So what was the regulatory status of that diagnostic at the time the drug was approved and the assay was being used to identify patients?
Yeah, it was a bit of a perfect storm. There was what's called an ASR assay that existed, analyzed specific reagent, which is a commercial fish. Okay. It was a commercial fish assay existed at the time that the ALK fusion was discovered, because anaplastic large cell lymphoma has been a known clinical entity for 10 years earlier. So the cytogenetics labs used ALK split apart fish for ALCL diagnosis for years. So there was an assay commercially available, not companion diagnostic for crizotinib, but the ability for Abbott to they had the assay on their shelf. They just had to repackage it as a companion diagnostic and go through the validation. So it was ALK was really quite a perfect storm.